Good afternoon, everyone. So welcome to APOL1 mediated kidney disease, a call to action with current knowledge and resources. This session is being supported by an educational grant by Vertex. And I'll introduce our speakers. My name is Bessie Young. I'm the Vice Dean for Equity, Diversity, and Inclusion at the University of Washington. It's my honor to be here. I'm a nephrologist and used to work at the VA, was section chief there. So our first talk will be The Biology of APOL1-Mediated Kidney Disease by Dr. Martin Pollack. And Dr. Pollack is the George C. Reisman Professor of Medicine at Harvard Medical School and the Chief of the Division of Nephrology at Beth Israel Deaconess Medical Center. His laboratory studies the molecular and genetic basis of kidney disease in humans, and his group uses genetic methods to identify and study the mechanisms of kidney disease genes. His laboratory is experienced in human genetic methods as well as cell-based studies of podocyte biology and the development of analysis of animal models of kidney disease. And he was very much on the forefront for APOL1, finding the gene. And so I will turn it over to Dr. Pollack.
Thanks very much, Bessie. It's a pleasure to be here. Let's see, and this is, okay, thanks. Forward, that works. I'm told by my institution I need to start with my disclosures. I have my laboratory has some intellectual property related to APOL1, and we received some research support from Vertex. So just to cut to the chase here, this is a picture from a paper we published now about 15 years ago, showing the really strong association between specific variants in the APOL1 gene and focal segmental glomerulosclerosis, FSGS, in people who identify as Black or African American. And you can see on the left-hand panel, there's a pair of single nucleotide polymorphisms, or SNPs, that are very highly associated with FSGS. When we control for that association, we still see a very strong association with another coding sequence SNP that changes the sequence of the encoded APOL1 protein. And when we control for both of these things, the association with this region of the genome goes away. So FSGS in African Americans is highly associated with this region of chromosome 22, and it's driven by these two alleles in APOL1, a pair of SNPs that are always inherited together, which we have come to call G1, and which changes two amino acids, and what we've come to call G2 allele is a 3 amino acid deletion in the C-terminus of APOL1. So what is APOL1? APOL1, well, we use the name APOL1 to refer to both the gene and the encoded protein, short for apolipoprotein L1. Apolipoprotein L1 is an apolipoprotein. It associates with lipid particles in the circulation, but it's also expressed in the kidney. Here is just a simple diagram of the basic structure of the APOL1 protein. Some isoforms of APOL1 have a signal peptide, others don't. It's followed by what's been called the pore-forming domain, although we now don't think that this region is actually encoding a pore, a membrane addressing domain, and what's called the SRA binding domain. I'll come back to what SRA is. The allele or variant that we call G1 is this pair of amino acid changes, serine to glycine change at residue 342, and isoleucine to methionine change at residue 384. And these are almost always inherited together. G2 is a deletion of 2 amino acids near the C terminus of APOL1, and this leads to an in-frame loss of 2 amino acids. Now, G1 and G2 are very close together. They're never inherited together on the same copy of the gene. And we have, as an easy kind of naming system, we've come to call anything that's not G1 or G2, we refer to it as G0. I guess I'm just wondering if the cursor works here. I guess it works there, but not there. Okay, pointer. That's so old-fashioned.
Yep.
So there are 6 basic genotypes a person can have. A person can have 2 G0s. A person can be heterozygous for G1 or G2, or have a risk allele from both parents. So be homozygous for G1, G2, or a compound heterozygous for G1 and G2. In the United States, about 50% of people who identify as African American or Black have a G0 genotype. About a third are heterozygous for G1 and G2. And then about 12-13% have 2 of these variant alleles. In general, most of the literature suggests that it's people with these 3 genotypes are the people who are at very high risk of kidney disease. There's increasing data, although it varies a lot from study to study, suggesting that there is a non-trivial risk of kidney disease associated with heterozygosity for G1 and G2. The important points here are that kidney disease is highly associated with having 2 risk alleles. APOL1, it's an apolipoprotein that circulates in the blood as part of HDL3, but it's also expressed in many tissues, including the kidney. And within the kidney, APOL1 is most highly expressed in the podocyte, and it's also expressed in the vasculature. APOL1 variants and these high-risk genotypes increase the risk of several forms of kidney disease, as we traditionally define kidney disease. On the far left are things where the odds ratio associated with these genotypes are extremely high. This includes things like interferon-associated FSGS. There are lots of case reports of people who receive therapeutic interferon who get FSGS. Almost everyone with that phenotype has a high-risk APOL1 genotype. HIV-associated nephropathy is, again, highly associated with APOL1 high-risk genotypes. In Africa, I think the best study has been done, the odds ratios are almost 90-fold greater risk of developing HIV-associated nephropathy if you have a high-risk APOL1 genotype compared to not having a high-risk APOL1 genotype in the setting of HIV infection. Odds ratios are very high, but a little bit less for focal segmental glomerulosclerosis. And in general, I won't go through each point on this slide, but as we go from very specifically defined forms of kidney disease to more general, loosely defined things like hypertension-associated kidney disease, these risk factor, the size of these odds ratios goes down. But what the point is here is really to hammer home the fact that a broad range of human kidney disease, defined in a lot of different ways, are highly associated with APOL1. And that's led to many of us really referring to a person who has a high-risk APOL1 genotype and one of these forms of disease is having APOL1-associated kidney disease or APOL1-mediated kidney disease, emphasizing the shared molecular genetic basis of disease. So I want to talk about APOL1 and its function. We can't really talk too much about function without talking about evolution, in my opinion. This is a kind of a complicated looking slide, but essentially it shows that the disease-associated alleles carry with it a larger range of haplotype in the genome than do the non-disease-associated alleles, indicated by the lack of these preserved bars, horizontal bars. That's a clue that something is going on here in terms of natural selection. Usually, when an allele is pretty common, like APOL1, the size of haplotypes around different variants are going to be quite small because recombination events through many generations have chopped that haplotype down. But the fact that it's quite large in the case of the APOL1 G1 and G2 alleles, the derived alleles, suggests that natural selection has selected for these alleles. There's been some selective advantage to having these kidney disease-associated genetic variants. And what is that? Well, this is straying a little bit from nephrology proper, but I think it's important to understand this issue of why are these highly deleterious variants so common. And this has to do with this parasitic disease, African sleeping sickness. So the etiologic agent of African sleeping sickness is Trypanosoma brucei. Trypanosoma brucei is pathogenic in most mammals, but it's not pathogenic in humans. And the reason for that is because normally APOL1 interacts, essentially kills Trypanosoma brucei. There are two subspecies of Trypanosoma brucei that cause disease in humans. Trypanosoma brucei gambiense, which causes chronic African sleeping sickness, and Trypanosoma brucei rhodesiense, which causes acute African sleeping sickness. And it turns out that G1 protects humans against Trypanosoma brucei gambiense. It's essentially, you can think of it as extending the protective effect of APOL1 against a broader range of trypanosomes. And G2 protects people against Trypanosoma brucei rhodesiense infection. And we believe that these variants arose in sub-Saharan Africa, probably 5,000 to 10,000 years ago, pretty recently, and well after the big out-of-Africa migrations that have populated our planet. So we really only find these variants in people with relatively recent African ancestry. And of course, that includes not just people in Africa, but many, a large percentage of people in the Americas, South America, and of course, in this country through the effects of the slave trade over the past 400 years. In Africa, these variants are more common in the West, less common in the East, and that corresponds to certain epidemiological observations. For example, HIV-associated nephropathy is much more common in West Africa. HIV is common in places like Ethiopia, but HIV-associated nephropathy is essentially non-existent there, consistent with what I told you earlier about APOL1 and HIV-associated nephropathy risk. So there's a really strong association with disease and APOL1 genotype. What's the mechanism of disease? What's going on? Why do these variants, why do these genotypes lead to what we call APOL1 mediated kidney disease? This is still something that's debated, but I want to talk about some of what we know about this. First of all, I think one of the most important points is that the kidney doesn't need APOL1. APOL1 is a very new gene in the genome. Our favorite experimental animals, mice, rats, for example, do not have an APOL1 gene. They have other APOL family members, but no APOL1. Our closest relatives, the chimpanzee, in fact, don't have an APOL1. Gorillas do, chimpanzees don't. So, you don't need, and this proves that you really don't need APOL1 to develop normal functioning kidneys. It's not needed for kidney development or kidney function. And it actually, this is a paper that appeared well before we knew any connection between APOL1 and kidney disease. A person was identified in a rural part of India who had an unusual trypanosome infection and had deletions in the APOL1 gene. Not amino acid changing point mutations, but deletions where he was unable to make any APOL1 at all. And again, this person had a weird trypanosome infection, but normal kidneys, normal kidney function. And we were able to track this guy down years later, still not even any microalbuminuria. So it really doesn't suggest that you need APOL1 for your kidneys to work. Another important question is the issue of why do only some people with high-risk APOL1 genotypes get kidney disease? The risk associated with kidney disease in people who have a high-risk APOL1 genotype is extremely high for variants this common. It's exceptionally unusual for common variants like this to have such a strong effect on risk of disease. But still, certainly not everyone gets disease. We estimate that probably the lifetime risk of developing overt clinically important kidney disease in people with high-risk APOL1 genotypes is probably 20-25%. So quite high, but not 100%. So why do some people get disease, others not? We think one explanation has to do with expression plus toxicity. We think these APOL1 variants, the variant forms, the G1 and G2 forms of APOL1 are toxic to cells in which they're expressed, particularly the podocytes. We also know that APOL1 is very upregulated. The expression, its RNA is very upregulated in response to interferons, like interferon gamma, as shown in this picture. Here, this is just a picture of the effect of of interferon gamma on APOL1 expression in podocytes and culture. On the right, this is just a simple experiment using HEK293 cells expressing the different variants of APOL1. The G0 form is a little bit toxic to cell. Oh, I'm sorry, a little bit toxic to cells. The risk variant forms are quite toxic. We think that what happens, at least in some cases of APOL1 mediated kidney disease, such as in these highly inflammatory states like HIV nephropathy, COVID-associated nephropathy, is that interferons and other cytokines upregulate APOL1. APOL1 is toxic to podocytes and perhaps other cells. This leads to overt disease. So it's toxic, but how is it toxic? And if you look through the literature, there have been a large number of papers addressing this question. I would say it's still not, there's still not universal agreement. And the proposed mechanisms, at least some of the proposed mechanisms are summarized here. A lot of data suggests that the risk variant forms of APOL1 can form a pore on cell membranes, and altered activity of this pore leads to cell lysis. Some studies have suggested that the risk variant forms of APOL1 enter the mitochondria and form a pore in the inner mitochondrial membrane. It's been suggested that the variant forms of APOL1 misfold in the endoplasmic reticulum, and this causes this process known as ER stress, and this is damaging to cells. And it's also been suggested that APOL1 risk variants directly alter the cytoskeleton of the podocyte through an alteration in an interaction with other APOL family members. So these models are out there in the literature. On I certainly favor the notion that APOL1 functions as an ion channel. This is a kind of a summary cartoon from a paper OPL published in my lab 10 years ago, just summarizing a bunch of experiments that suggested that the variant forms of APOL1 have increased cation channel activity, loss of potassium, and a lot of downstream toxic cellular consequences, which I won't review in any detail. But I'll say there's really a lot of data from many labs that suggest APOL1 functions as an ion channel. This includes work in trypanosome membranes, where we think it's the ion channel activity of APOL1 that kills the trypanosome, lyses it. Studies where APOL1 is expressed in xenopus oocytes, HEK cells, lipid bilayers, liposomes, zebrafish, podocytes. A wealth of data in a variety of experimental systems suggests that APOL1 can function as an ion channel. This is just one hypothetical schematic of what this transmembrane structure of APOL1 might look like, with what we think is the ion channel pore, in fact, near the C-terminus of the protein. So I certainly think that the bulk of data suggests that variant APOL1 has altered, probably increased ion channel activity at cell membranes, and this is what's pathogenic in terms of disease. But it's not quite that simple. So for example, we've found that the risk variant forms of APOL1 interact with each other differently than the G0 or wild type form. We see many more of these high molecular weight oligomers form. And this is a blot from cell lysates of HEK293 cells. We've seen something similar when we look at other ways of expressing APOL1. But G1 and G2 seem to multimerize or oligomerize differently than G0. Whether that is also an independent pathologic process or perhaps a mechanism by which APOL1 variants have increased channel activity or just a epiphenomenon, we don't know. The regulation of APOL1 expression is pretty complex. This is just a figure to illustrate one aspect of this. APOL1 has in the 3' region of the genome these what are called ALU repeats, these highly repetitive regions of the genome that tend to be highly edited by enzymes known as adenosine deaminases. And again, it's not super important that you remember this, but the point is that ADAR, like APOL1, is highly induced by interferon. ADAR can edit the 3' UTR of APOL1 and decrease its expression. So there's complicated feedback loops involved in the expression of APOL1. So, I kind of like to think of it sort of as this kind of cloud of complex interactions between factors. And these include factors intrinsic to to the organism, the human being, and external factors, factors in the environment. So at the level of things that are part of the organism, part of the human being, things in the genome that affect disease, the genotype of APOL1, potentially the haplotype it sits on, various aspects of regulation of the transcript expression, perhaps post-translational modifications of APOL1, which are we and others are studying, and function of the protein, where it localizes in the cell, what its topology is, how it dimerizes, and so forth. But there are clearly environmental factors also that are critically important. Infections such as HIV, COVID-19, and probably others that we haven't identified are likely important in triggering overt APOL1-mediated kidney disease. And these whether the phenotype we're talking about has very high odds ratios associated with disease risk or lower, probably depends on the nature of largely these environmental stimuli. What's triggering the development of overt kidney disease? Is it HIV infection? Is it COVID-19 infection? What's going on with diabetes is something that's unclear. Some studies have suggested that the rate of incident diabetic nephropathy is not different as a function of APOL1 genotype, but it does seem clear that people with diabetes, established kidney disease, their disease progresses more quickly to end-stage renal disease as a function of APOL1 genotype. Less clear that it makes you more likely to develop diabetic nephropathy, but it does seem to make CKD in the setting of diabetes progress more quickly to kidney failure. So one of the things that we like to do in the laboratory is make models of disease. So I'll be very brief and I'll just tell you about a model we made. A lot of people have made models of disease. We did it by making a back transgenic mouse. So mice don't normally have an APOL1, so we have to put the human gene back into mice. And we created mice with G0, G1, or G2 transgenes all inserted into the same place in the genome. And they have no phenotype. When we do this, there's no phenotype at all. But and at baseline, we see no significant expression of APOL1 in the kidney. We see a lot of nephrin as a control here. But when we treat with interferon gamma, when we deliver interferon gamma to these mice, they develop a lot of APOL1 expression in the podocyte. They get a lot of proteinuria, as you can see here, whereas the G0 mice develop no proteinuria, and the genetic controls for these studies develop no proteinuria also. So although in humans, it's very complex, not everyone with high-risk genotypes gets disease. When we isolate these genetic variants, put them into mice and treat them with this particular stimulus, we see what's essentially like a Mendelian disease. Everyone with all these mice with their G1, G1, or G2, G2 get severe proteinuria. None of the mice that are G0, G0 do. So if you have any lingering doubt as to whether this is merely an association of a variant with disease as opposed to being causal, I think this really proves that these variants cause human kidney disease. And just to show you, for those of you who like to look at kidney pathology, again, the G2, G2 mice and G1, G1 mice, we see severe glomerular sclerosis, which we don't see in the G0, G0 mice. I'm going to skip this because I want to stay on time. What are the key questions about APOL1 biology? I think one of the most interesting questions has to do with what is the largely recessive nature of disease. As I mentioned earlier, some studies, including a recent study by the H3 Africa Consortium, showed a significant risk associated with heterozygosity for G1 or G2. But for the most part, really, the risk seems much greater in people with two risk alleles. Certainly in our mouse model, we don't see any risk of kidney disease in our mice that are heterozygous for G1 or G2. So this leads to questions of loss of function or gain of function. Usually recessive diseases we think of as being loss of function diseases, as being early child onset diseases where you don't make a protein and you have early manifestations of disease because you make none of the protein. Gain of function diseases we think more typically of dominant diseases where the mutant has some added activity, the mutation confers some toxic effect. And so, one's good enough to be toxic. So this is a little bit of a puzzle. We don't really understand why penetrance and phenotype are so variable. And again, this question of if these are gain of function toxic variants like I mentioned, why is it largely recessive? I will skip this. I will just mention this is just a recent picture from studies in podocytes from cultured from the mice that I showed you. We see increased cation channel activity in podocytes from G1 mice compared to mice of other genotypes, suggesting that the channel changes we see in very artificial systems are probably real. I also will mention, one last thing. By mistake, we developed a APOL1 G2 transgenic mouse with 28 copies instead of 1 copy of the mutant APOL1 gene. And these mice also don't have any kidney disease at baseline. It's only when we stimulate them with interferon do we see overt disease. That raises the question of whether just high levels of expression of APOL1 alone are sufficient to cause disease, or if there's other aspects of the inflammatory milieu that are important. This is among the questions we're trying to answer now. One more slide. I think you might hear a little bit more about this later, but I just want to point out this observation about kidney transplantation. It seems to be the case that when we look at genotypes of kidney transplant recipients and donors, transplantation fares better using kidneys, deceased donor kidneys that are APOL1 low-risk genotype than high-risk genotype. That has a big impact on how well the kidney does. The recipient genotype seems to have less of an effect. There's been some debate about if there's any effect or there's a small effect of recipient genotype. The point is, disease risk mostly goes with the kidney. And in addition to clinical implications, which I won't talk about, this has biologic implications, right? It says that it's not the APOL1 in the circulation that's causing disease, it's the APOL1 expressed in the kidney. So in summary, we think APOL1 kidney disease is caused by gain-of-function mutations in APOL1 expressed in the kidney, particularly the podocyte, that lead to increased cation channel activity. And I'll end there, and happy to take questions if there's any time.
Thank you so much for that wonderful talk. I think we have time for maybe one or two questions. And I'll start. You showed your mouse model that replicated APOL1 kidney disease. And I read a paper where you actually gave lisinopril and it decreased the kidney disease. Can you speak about that?
Yeah. So we did a study where we gave lisinopril to the G1 and G2 transgenic mice, and it's pretty effective at reducing proteinuria or preventing proteinuria in the G1 mice, but not at all in the G2 mice. I don't have a biological explanation for that. I think it certainly suggests, again, a few things. It strengthens the notion that these are gain-of-function mutations, not because G1 and G2 are doing different things. They're not just ablating the function of APOL1. And it also suggests these maybe there are differences in the biology of disease as a function of genotype, and we need to look more closely at this when we genotype our patients, not just call people high risk or low risk, but really knowing G1, G2 might matter.
Thank you. Use the mic if you have a question. One more question. Quickly.
Thank you very much for the elegant presentation. I just want to know the sequence of event in clinical purposes. So I work in South Carolina where most of my patients are African American. So you suggest to screen people for APOL1, and if they are positive, do the genetic testing, or if they are positive, you don't do the genetic testing since they are already positive?
Well, I mean, the APOL1, it is a genetic test.
No, I'm talking about G1,
right.
G1, G2.
Right. I mean, so a lot of the panels now, the genetic panels that people do, they will tell you if it's G1, G2, G0. So I think that's a one-step process now. I think that, so you raise a really interesting question. In people, should we be screening people who are healthy to know their APOL1 genotype? And I don't think the answer is clear. I mean, one could make an argument that maybe we should be screening general population more for albuminuria. I'm a believer in that, something the primary care physicians don't believe in. But I think, identifying people with kidney disease early is important. And then maybe if we identify someone with early evidence of kidney disease, we might then genotype those people. That's the algorithm I would favor. There's another question that comes up, and I think is also, it's tricky, is that, these variants, of course, these are related to ancestry, not race. But there's, of course, there's an association between race and ancestry. But should we, not everyone with, most people who identify as African American or Black don't have these risk genotypes. So one shouldn't make the assumption that a person who is Black, has kidney disease, has APOL1 kidney disease, that could mislead you into missing a diagnosis. The other issue is, of course, should we be genotyping everyone irrespective of their racial identification? Because most people don't know the details of their recent ancestry. People barely know who their great grandmother is. So I think there's a good argument to be made for screening everyone, especially given that genetic testing and is becoming easier and faster and cheaper.
I think it would be fair to screen every person with proteinuria. I think that would be fair.
Yeah, I don't disagree with that.
Thank you so much. And that is a perfect segue. One more clap for Dr. Martin Pollack. That is a perfect segue into Natalie Vina. Natalie is a genetic counselor in the Division of Nephrology at Columbia University Medical Center. She's engaged in clinical and research efforts and is focused on identifying and understanding the genetic causes of kidney disease. She is an assistant professor of genetic counseling and is involved in training future genetic counselors, counseling professionals at Columbia University. Welcome.
Hi, everyone. Good afternoon, and thank you for inviting me here today to speak about APOL1 genetic testing, ancestry, and considerations in clinical care. So my name is Natalie. I am a research and clinical genetic counselor at Columbia, and I have no disclosures. And for the last 5 years, my focus in the genetic counseling space has really been in genetics and genetic testing of patients with various kidney diseases. And so today, I'm going to be discussing APOL1 testing from that perspective. And so today, I'm just going to go over a little bit about the clinical significance of APOL1, the high-risk genotype, discuss who are appropriate candidates for testing, and then provide some insight onto how to approach genetic testing for these variants, and then address some of the clinical considerations for implementing this testing both equitably and ethically. So I know we've heard about this a little bit earlier, but just for context and nomenclature, just some brief background here. When we think about the APOL1 high-risk variants, high-risk variants, we're really referring to two genetic alleles here referred to as the G1 and G2 haplotype. And these alleles evolved over 10,000 years ago in Africa due to their protective effect against African sleeping sickness, which led to positive selection over time and then result in this increased prevalence of these alleles in certain sub-Saharan African populations. And when we think about the high-risk APOL1 genotype, we're referring to the presence of the two risk alleles, so either G1 homozygotes, G2 homozygotes, or G1, G2 compound hets. And it's this biallelic combination that we refer to as the high-risk genotype that confers this risk for kidney disease. And so as we heard, the prevalence of these genotypes does vary widely worldwide and can present significant challenges when we're trying to identify our at-risk populations. So these two alleles did arise out of Africa, and while they are still prevalent there today due to historical population movements, particularly the African slave trade, we do see these variants at higher frequencies in the United States, the Caribbean, and as well as other of South America as well. In the United States, the G1 and the G2 allele can be found in roughly 22% of and 13% of Black individuals, respectively, which then translates to about 13% having this high-risk genotype. In the Caribbean populations, such as Jamaica and Barbados, this can be seen around 10%, but can get up to as high as 22% in specific areas. And then we also heard too earlier, the H3 Africa Kidney Disease Research Network, they found recently a staggering 43% of their populations within Africa have a monoallelic form of the variant, whereas on almost 30% are biallelic. However, even within West Africa, they noted significant variability in the prevalence of this risk allele, again, within different linguistic and ethnic populations. And as sure we've all seen this slide before, but I do think this slide, as well as the previous point, just highlight that even though there's a high prevalence of these risk alleles, there is challenges in identifying those with the high-risk genotype, especially in those where the genetic ancestral origins are complex and diverse. So now that we know clinically or genetically what the high-risk genotype is, what does this mean clinically? And clinically, what this means is that an individual with a high-risk genotype has a higher chance of developing kidney disease than someone who does not at its basic level. Specifically, they are more likely to develop hypertension-attributed ESKD, lupus nephritis ESKD, FSGS, and HIV-associated nephropathy. And so collectively, any non-diabetic kidney disease in an individual with a high-risk genotype, that's collectively known as APOL1-mediated kidney disease, or AMKD. And to quantify this risk a little bit, individuals with a high-risk genotype have around a 15 to 30% lifetime risk of ESKD, which is about a 7 to 10-fold increased risk compared to those without those variants. Similarly, biallelic APOL1 variants are associated with an increased odds ratio of about 7 to 17% for FSGS, and that is estimated that approximately 75% of Black individuals in the United States with FSGS actually have a high-risk genotype. And for the individuals who do develop AMKD, the clinical burden is pretty substantial. It's been demonstrated that they have a faster time, a more aggressive course of kidney disease with a more rapid time to rapid decline in EGFR and a more fast time to ESKD than those without the high-risk genotype. And while the burden of disease is typically attributed to the biallelic variants in an autosomal recessive inheritance pattern, there is some evidence emerging out of the H3 Africa study that found that a single risk allele can be associated with an increased risk of, or an odds of CKD of 18% and FSGS of about 61%. And while more information is needed to, understand this risk further, this can change dramatically how we think about these risk alleles. When thinking about the APOL1 testing in the transplant setting, these genetic changes do have important information and risks for both donors and recipients. Evidence does suggest that in individuals who receive a kidney from a donor with a high-risk genotype, they have shorter allograft survival rates compared to those who have a donor with a low-risk genotype. And then the donors themselves with a high-risk genotype, they can demonstrate a greater decrease in kidney function post-donation and a higher risk of developing ESKD themselves than those without the high-risk genotype. And although these associations are evolving and they're the subject of several ongoing studies, such as the APOLLO study, I think this study is aiming to better characterize the risks associated with for both donors and recipients. So going back to AMKD, it is critical to emphasize that upwards of almost 80% of individuals with a high-risk genotype never develop kidney disease. And this points to the impact of disease modifiers. We know that there are modifiers that can both increase and decrease the risk of developing kidney disease with a high-risk genotype, such as the protective N264K variant that mitigates the risk that is conferred by the G2 allele, as well as we know that there are inflammatory and infectious triggers that can accelerate or initiate disease presentation. And so without going into further detail about what all this means, I do detail about it. What it means is that when we think about genotype and phenotype correlations in AMKD, it's very variable, which makes it very difficult to identify individuals with a high-risk genotype based on clinical presentation alone. And this variability can lead to inconsistent and inequitable access to genetic testing for those and the at-risk population. And it raises the critical question over who should be tested, how do we identify these populations, and when is it appropriate to do this in clinical care? So to answer this question, I do have to provide some framework and just general background in genetic testing. So generally, when we think about clinical genetic testing, we tend to think about it in two main groups, diagnostic and predictive testing. So diagnostic testing is performed in an individual with symptoms and is used to identify or confirm the genetic cause for said symptoms. So in the setting of AMKD or any symptomatic patient, APOL1 genetic testing would be considered diagnostic testing. As such, APOL1 testing should be considered for any individual with sub-Saharan African ancestry who presents with non-diabetic kidney disease, particularly those with FSGS, HIV, nephropathy, and maybe even lupus nephritis. Predictive testing, on the other hand, is genetic testing of an asymptomatic individual to determine their risk of developing a condition in the future. And so broadly speaking, in the setting of APOL1 predictive testing, due to the low penetrance of the phenotype, the limited prevalence of the high-risk genotype in those with non-African ancestry, as well as the current lack of treatment and disease prevention, the clinical utility of predictive testing in this space is limited and not really, at this point, recommended for broad population-level screening. Now, that is not to say that predictive testing has no utility at all or won't have utility in the future. For instance, there are emerging studies that suggest that early identification of a high-risk genotype prior to the onset of AMKD may be associated with decreased blood pressure, better adherence to medication, and positive lifestyle changes. But nevertheless, at this point, based on current evidence, testing of asymptomatic patients for APOL1 risk allele should be approached with caution in the setting and take into mind the clinical circumstances. There is one notable exception, however, when thinking about asymptomatic testing for APOL1, and that's as it relates to kidney donors. So as noted earlier, although this information is still evolving, current evidence does suggest adverse outcomes for both the donor and recipient when they have a high-risk genotype. As such, in 2017, KDIGO recommended considering APOL1 testing for donors with sub-Saharan African ancestry, and the American Society of Transplantation recommends that all living donors be informed of their risk associated with APOL1, the high-risk genotype, as well as availability of genetic testing. But both of these recommendations stop short of universally offering or recommending universal donor screening. So in clinical practice, this translates to pretty significantly different and variable APOL1 testing policies across institutions. There are several ongoing efforts with key stakeholders to harmonize and address these inconsistencies and guide policy recommendations. But what has emerged from these discussions is that while most do support universal offering of APOL1 genetic testing to potential kidney donors of sub-Saharan African ancestry, they highly recommend a shared decision-making model be utilized when deciding to whether or not to actually undergo testing, and then how that information is used in clinical decision-making in the transplant setting. And what this means is that no one person should unilaterally make a treatment decision, but rather with proper consent and informed decision-making, APOL1 testing, or lack thereof, is just one of the many aspects when thinking about the best clinical scenarios for both the donors and the recipients. Which brings me to the considerations for implementing APOL1 genetic testing in the clinic. Now, I do need to take a step back and acknowledge that broadly speaking, and at its basic level, APOL1 genetic testing is focused on individuals of a particular ancestry. And in this case, it's the sub-Saharan African ancestry. And thus, by definition, it constitutes race-based or race-targeted testing. And while this type of testing is not new or unique to this particular ancestry, this type of testing does present a unique set of challenges and special considerations when thinking about test implementation. For starters, it's important to acknowledge that race and ethnicity are social constructs and fundamentally different than genetic ancestry. Relying on race to infer ancestry is not only imprecise, but inaccurate and been shown to be inaccurate across several studies. And while it's widely recommended that self-identified race and ethnicity, and so it is widely recommended that self-reported race and ethnicity not be used as proxies for genetic ancestries in clinical decision-making. And as noted, the APOL1 high-risk variants are found almost exclusively in individuals with sub-Saharan African ancestry. And due to the migration patterns and admixture, there is considerable ethnic and cultural diversity within this group. Therefore, when considering who should be offered APOL1 testing, providers must avoid making assumptions or inferences based on perceived race. For example, in the United States, individuals with African ancestry can identify as Black, African American, Caribbean, among several others. And so the most accurate and equitable way to identify at-risk populations is to directly ask patients if they have African ancestry. And any individual who does identify as having African ancestry, who also presents with non-diabetic CKD, should be offered APOL1 genetic testing. And while this approach is definitely not exact and does rely on patient report, it provides a more equitable access and equitable way to offer APOL1 genetic testing. It's also important to anticipate and address potential barriers to widespread genetic testing for APOL1 within this particular ancestral group. As such, it's critical to involve key stakeholders when developing policies and programs around genetic testing so that we're not unintentionally creating new forms of exclusion and we're overall promoting health equity. For instance, while there may be valuable prognostic information to gain in APOL1 testing, there are concerns that testing, universal testing, could create additional barriers to living donation for Black patients, which can in turn further exacerbate the existing health inequities that exist. And so to mitigate these risks, equitable and informed offerings of genetic testing for APOL1 is essential. And so this then raises the importance of informed pre- and post-counseling for APOL1 genetic testing. So pre-test counseling should provide a clear explanation of what the APOL1 high-risk variants are, as well as the risks that can be conferred with them, including both the potential implications of both a positive and negative result. These discussions should be tailored to the patient's specific circumstances and within their clinical context, which can differ significantly depending on the disease status of the individual. For example, in a patient with existing kidney disease, the pretest counseling may focus on clarifying their diagnosis, identifying the etiology of disease, informing prognosis, and discussing potential implications for relatives. In contrast, for asymptomatic individuals, particularly those with a family history or those undergoing transplant donor evaluation, counseling should emphasize their risk assessment, decision making, and the potential of outcomes that could happen in the setting of a positive result. Post-test counseling is equally as important as well. When returning a high-risk result, it's important to contextualize these findings within the patient's experiences as well as their motivations for undergoing testing. For some, a high-risk genotype could bring clarity to a disease etiology in a symptomatic patient, for example, whereas for others, it may raise uncertainties about their future risks for developing kidney disease, as well as introduce some uncertainty in the transplant process. For individuals with a high-risk genotype, cascade or familial testing may be appropriate and should be discussed with the patient, particularly as it relates to familial transplantation donation. And lastly, decisions if and when to test children, particularly asymptomatic children, should balance the potential benefits of risk stratification as well as their autonomy. Now, ultimately, as providers, it's important that we engage in culturally sensitive genetic testing that's grounded in equity. And the goal for the genetic counseling is one that promotes patient-centric informed decision-making and provides support for those considering or undergoing APOL1 genetic testing. So I do just want to switch gears a little bit and talk about some practical testing considerations for genetic testing as well. So first is test selection. There are several commercial options available for testing these days with advantages and disadvantages to them both. So for instance, targeted testing of the 2 risk alleles is accurate and rapid, but there may be missing out on other monogenic modifiers or diseases that would be detected with larger multi-gene panels. And similarly, cost is often a perceived barrier to testing, but based on clinical experience, there are several options available to patients to help them gain access to testing with minimal financial burden. So this includes things like fighting for insurance coverage, the lab's particular billing policies themselves, financial assistance, as well as other like no-cost sponsor testing. All of these can help offset the potential financial burden to genetic testing. And as always, there are privacy concerns to consider. So I would be remiss without mentioning GINA in a talk about genetics. So GINA, the Genetic Information Non-Discrimination Act, is a federal law which protects asymptomatic or patients at risk of developing a genetic condition against discrimination from healthcare and employment. But as with most laws, there are limitations and caveats. So for instance, GINA does not apply to other forms of insurance, such as disability insurance or life insurance. And second, GINA only applies to those who are asymptomatic for the given condition. So specifically applies in the setting of predictive testing. And most symptomatic individuals are not going to be covered by GINA. There are other federal laws that can protect them, but not GINA. And while there are, again, these other considerations or things, it's important to think about in this particular patient population because over 80% of our patients with the high-risk genotype will never develop symptoms of their condition. And so GINA is particularly important to consider with this group. And so on that note, before I end, I do have a poll here. And so I am just going to note that I am asking you to make an assumption, which I explicitly said not to do, but for the sake of polling, I can't really ascertain that. So here are a couple clinical scenarios, and I just would like to know which one of the following would be offered APOL1 testing, and there should be another option at the bottom that says all of the above, which is not here. Oh, there it is. Okay. So I'll just give you guys a minute. I think I gave it away a little bit, though. Sorry. Okay, so in the interest of time, I am just going to jump forward and just say that, yes, the answer was all of the above. And even though I asked you to explicitly not make those assumptions, with the proper counseling and informed decision-making with the patient, then yes, theoretically, all of these patients should be or could be offered genetic testing for APOL1 based on their current scenarios. And with that, I will conclude. But I do want to actually just take one moment and just stress that APOL1 testing in this patient population, it does provide us a very, very powerful opportunity to understand and manage kidney disease risk in this very particularly disparaged patient population. But by promoting informed decision-making and offering equitable testing, access to testing, I hope that we can then support the appropriate and responsible use of APOL1 testing in clinical care for this population. So now I will be done with questions.
Thanks, Natalie. So I'll open this up for questions. There was an anonymous question that I'll just read. It's about genetic testing. So what is the guidance on testing for individuals who are of mixed race with African ancestry, even if they don't identify as Black or African American? And I think this is a very important question. I'll expand it to, if we do a kidney biopsy and a patient has FSGS, what are the recommendations for testing for APOL1?
Yeah, so I think that's a good question. Here, as I mentioned too, the goal, I think, is to think about equitable access and not just thinking about it which does then bring us to the concept of thinking about it in our particular patient population. And on that note, I do think that there are specific clinical indications where genetic testing is warranted, but a broad approach to genetic testing should be considered as well. So most of our panels these days do include APOL1 as a gene of interest. And so instead of thinking it in the clinical context of specific genetic changes, think about it more broadly in terms of genetic testing in that regard. So that would be my answer. Think about it more broadly versus more specifically, especially when we are thinking about admixed populations.
Thank you. I'll go with.
Thank you. Thanks for the wonderful talk. I'm Ehsan Novak from George Washington University. There are some genetic variants that are called likely pathogenic or pathogenic, and they do not have full penetrance, as you mentioned. And at what point you call APOL1 likely pathogenic?
Yeah, great question. So this is actually quite a hot topic, I guess, in our field. Where does the line cross between having it to be disease-causing versus a risk factor? And I think as Dr. Pollack showed, these variants themselves do cause disease, be it at a reduced penetrance. So I do think that there is a little bit of a misnomer here that these are variants that are actually conferring disease when thinking about it from an environment it's just not a single genetic cause that's causing it. It's like a multi-hit model, but it is the presence of these variants that is causing disease in those folks. So I do think that in terms of nomenclature, right now, these are called risk variants, but I do think that's going to change over time. I do think that these are going to end up their nomenclature is going to change as more evidence on the biology of them comes to light.
And thank you. And how do you counsel a patient, a donor for transplant with just one risk allele?
Also a great question right now. The evidence over whether or not there is substantial risk is still evolving. So right now, in clinical practice, when you only have one risk allele, we are treating them as not at having like a much more higher risk. I think the evidence is still coming out, and the studies are needed to further support that claim that the heterozygotes do have risk, and to better quantify that. But until we have that data, right now we're really only counseling about the high risk with the biallelic genotype.
There was one question about, do you believe that kidney donors should be screened for APOL1 genotype?
Do I? I think.
Or do?
I think as long as we are doing it in an equitable way, yes. That means access to testing for everyone everyone with the proper counseling. So yes, I do believe that donors should be offered testing and counseled on testing, but I don't think that they're necessarily, I think the decision to undergo testing is personal.
Maybe just one more question. The question is, I guess, Why exclude people with diabetes who may have sub-Saharan genetic ancestry from recommendations for genetic testing?
Yeah, right now, I think that the evidence is just that there's not a huge risk associated with that phenotype, and that I should say that the risk alleles are not conferring that risk for that particular phenotype. And that's the only reason why.
And then this last question, maybe I'll ask you about, do you think that patients with APOL1 gene could have another cause of CKD, may still have the APOL1 mutation but not express the disease?
Yeah, that's an interesting question. So, I mean, certainly someone with a high-risk APOL1 genotype can, develop acute kidney injury or ANCA-mediated vasculitis or all these things. So, it certainly doesn't preclude some other mechanism being the main driver of kidney disease. I think, I do think that for most kidney diseases, however they were initiated, progression to kidney failure is probably going to be greater in people with these genetic variants. I think there are plenty of kidney diseases where APOL1 genotype does not contribute in a direct way to pathogenesis, if that's the basic question here.
All right, thank you so much for that talk. All right, I have the final talk, and some of my slides overlap, so I will breeze through those because you are now all experts on APOL1 mediated kidney disease. Okay, so my talk is APOL1 mediated kidney disease, current management, clinical outcomes, and emerging therapies. So I will give a little bit of a background and quickly go over some of the slides. I'll spend a little bit of time on the epidemiology and also have a few questions for everyone. In my role now as the Vice Dean for Equity, Diversity, and Inclusion, I always start at the Office of Healthcare Equity at the University of Washington. I kind of start my slides with honoring the land on which the UW stands, and we honor the Coast Salish tribes. They're people, the land on which touches the shared waters of the tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And I also want to acknowledge that we are all living on land that was ancestral lands of indigenous peoples for thousands of years, and we acknowledge the extraction of brilliance, energy, and life for labor forced upon the people of African descent for more than 400 years. There's also, to this I add, there was a force of labor for those of Asian descent as well. So indigenous people and descendants of Africa have shown in this country and worldwide that they have the resilience and strength, and we carry all of our ancestors with us, and we are continually called to be better as we lead this work. I also have a disclosure that I am the site PI for our Vertex study, but I don't receive any financial compensation. And I'll start with our objectives. So the first is to review the link between APOL1, apolipoprotein L1 and kidney disease and how that was made. And you've heard from the person who actually made that link. I'll review some of the epidemiology of APOL1 kidney disease, current evidence-based treatments for APOL1 kidney disease, which many of you are familiar with, but we'll go through what I could find. Discuss emerging therapies for APOL1 kidney disease, and then go to conclusions. So I'll start with this case. I'll have you go ahead and read this and then think about what your answer is. 42-year-old African-American male with a history of hypertension presents with progressive proteinuria and declining kidney function. He has no history of diabetes, and a kidney biopsy shows focal segmental glomerulosclerosis without significant immune deposits. Genetic testing reveals 2 high-risk alleles for APOL1. Which of the following best describes the pathophysiology of this condition? All right. Everyone. So sounds like everybody was paying attention. That's good. So we think it's direct podocyte toxicity leading to increased cell injury and apoptosis. So I'll start. So you all know this. Chronic kidney disease affects about 37 million Americans at a cost of about $37 billion in end-stage kidney disease care in the U.S. African Americans and Black individuals have a 2 to 4 times greater risk of developing kidney failure or end-stage kidney disease compared to whites. And white individuals and African American individuals comprise about 13% of the population, but about 35% of dialysis patients. And in places where there are more African Americans, like the South, it's probably like 80 to 95% of dialysis population. So we know this. I also put this in just to remind us of the incidence of end-stage kidney disease is much greater in some of our racial and ethnic minority populations. And this slide has always fascinated me that those who identify as Black or African American have always had this much higher risk of kidney disease and end-stage kidney disease compared to other races and ethnicities. And this is another slide that's a little bit dated, but when you look at when kidney disease develops, it develops earlier, about 10 years earlier, and then sort of really rises up very quickly. So there is this increased risk. We know that there are a lot of risk factors that contribute to this. So hypertension, diabetes, family history of kidney disease, smoking, poverty, and lack of access to care. And cardiovascular disease. And many of these overlap with sort of the genetic environment. I also want to bring in social determinants of health because we know that people need to have access to care in order to have their hypertension treated and their diabetes treated, which is so important. And sometimes by the time we see them as nephrologists, they've already developed kidney disease, which probably could have been prevented at some point. So this is the paper that I always refer to, and Dr. Pollack and his lab found this association of APOL1 variants with kidney disease in African Americans. And this is what's from ASN Kidney News way back in 2010, where you can see the trypanosomes in sort of the red blood cells together. So I, again, will quickly go through this because you've heard about it. So trypanosomes are the parasites that are endemic in Africa. They're transferred from the gut of the tsetse fly to humans. They live inside the human body and multiply rapidly and cross the blood-brain barrier to cause African sleeping sickness. And these variants arose some 3,000 to 6,000 to probably 10,000 years ago in Africa and really are associated with the resistance to the lethal form of African sleeping sickness and sort of spread by the tsetse fly. And you've seen this picture of this slide that looks at APOL1 variants that are associated with non-diabetic kidney disease. So as Dr. Pollack said, G1 has 2 amino acid substitutions, G2 has 2 amino acid deletions, and G1 is present. And these were data when this first became available. G1 was present in about 40% of Yoruba individuals in Nigeria. And I'll show you some of the data from H3 that actually goes into a little bit deeper evaluation of the epidemiology. And it's not present in those who are of European, Japanese, or Chinese descent. And it is thought, based on what we knew back then, that about 30% of U.S. African American or Black individuals have either G1, G2, or both G1 and G2, and about 13% of African Americans or Black individuals have both G1 and G2 and have the high-risk variants of APOL1. And as we've been discussing, it's thought that APOL1 is an autosomal recessive trait. I think we've gone through this. So I just wanted to spend a little bit of time on this, some of the older data. And I have their normal, but that's really the wild type of APOL1. So the initial studies were these case control studies where what was looked at was the genotype and basically the case control study of end-stage kidney disease. So if you looked at people who had end-stage kidney disease associated with focal segmental glomerular sclerosis, they had like a tenfold greater risk of having that association. For hypertensive kidney disease, it was 7.3. fold risk, and for HIV nephropathy, it was 29-fold risk. So again, pointing to what Dr. Pollock discussed. And in transplantation, there's been a variety of data that have shown that the risk of APOL1 is really associated with the donor kidney. So after these initial case control studies, there are a lot of population-based studies that looked at the odds of developing kidney disease, maybe defined differently, like whether or not somebody had CKD or albuminuria or rapidly progressed to kidney failure, where they included people from a broader population. And so those odds ratios or hazards ratios, depending on the study, decreased significantly. So now when you look at the more recent sort of population-based studies, so the risk is about twofold, 2 to 3-fold, depending on the population. We did some work with APOL1 and albuminuria in the Jackson Heart Study. So the Jackson Heart Study is a study of African Americans in Jackson, Mississippi, people who identify as African American. There's about 5,000 of them, and we looked at whether compared to 0 alleles, if a person had 1 allele of the high-risk variant or 2 alleles, whether or not they developed incident albuminuria or incident dialysis or incident chronic kidney disease or a decline in eGFR. And I think because the study probably didn't have enough of a population to look at the 1 allele, 2 alleles always had this greater risk of either albuminuria. There was a 9-fold greater risk of dialysis initiation and a 1.6-fold incidence of CKD, and then a 2-fold incidence of decline in CKD. So these are all just very important findings just looking at the epidemiology of APOL1 in U.S. populations. And so this gets us to transplants and donors, and we've heard some information about this, and I just replicated a table from Barry Friedman, who had put together this table of what we know about disease risk in donor and recipient APOL1 high-risk genotypes on kidney transplantation. So the first studies were done back in 2011 that showed that decreased donors had faster graft failure with APOL1 high-risk genotypes. That was replicated basically in 2015 and 2016 by Barry. That both were retrospective studies. Then he looked at deceased donor kidney transplant recipients, and this was a study by Lee that showed that recipient genotype did not affect the outcome. And so that's where we got that, recipients who had APOL1, if they needed to really have the kidney from the donor that had APOL1. There is a paper by Chen that looked at donors and recipients, and the APOL1 donor kidneys had 15% fewer podocytes and had more rapid kidney function decline in the recipients. So looking at sort of podocyte number. And then more recently in 2021, Zhang looked at donors and recipients and found that recipients with APOL1, either 1 or 2 hazard rate high-risk variants had increased graft failure and T-cell mediated rejection of the graft. So I think that this information is interesting. I defer to Dr. Pollock to describe that interaction a little bit more, but I think the findings are very interesting. And as we get more information, we'll be able to determine more about the actual mechanisms. So as was discussed before, this is a slide that just tries to show how APOL1 sort of came to North America and South America, this global migration and prevalence of the different genotypes. So there was the global migration about 10 to 20,000 years ago. There was the slave trade that brought enslaved individuals both to South America, to the Caribbean, and to North America. That is why we have sort of this area where there's increased APOL1 in the Americas, and then we have sort of the increased risk in sort of West Africa or Sub-Saharan Africa that is not in East Africa or not in Asia. And this is also a slide that you saw where this investigator took basically different studies and looked at APOL1 and saw that there was this higher risk of APOL1 in African Americans, also seen in West Africa, and then was sort of the first to show that there was more increased risk of APOL1 in the Caribbean. So very interesting sort of genetics. The next slide, I'll just show that APOL1 high-risk genotype distribution among ethnic groups in Africa. So this is some of the newer data. This is the data from H3 Africa, where they looked at the different ethnicities in Nigeria and Ghana. And you can see here that in Ghana and Nigeria, that there's sort of a difference in the prevalence. So it's higher in some ethnicities and lower in some that probably follow a geographic distribution, but very interesting in terms of the information regarding how APOL1 is showing up in Africa. And some other clinical data from this study that was just published in the New England Journal shows that there are, go back. If you have 2 copies of APOL1, you have this 25% risk of chronic kidney disease. So they looked at chronic kidney disease. Most of these were biopsy proven. They looked at whether you had just one G1 or one G2 variant and the risk of chronic kidney disease, and showed that there was risk with either G1 or G2 that was significant. They also showed that if you looked at the G2, G2, that had the highest risk. So that's where we're starting to see that there are differences in whether people are heterozygote or sort of they have just G1, G1, just G1, G2, or compared to G0 and G0, and whether or not they have just G2. So very interesting. And this paper also looked at the monoallelic and biallelic APOL1 variants with chronic kidney disease and looked at biopsies. And I just put this in to show that there is this very high risk of FSGS in the folks who had G2 variants, so that it seems to be consistent. And they also showed that if they looked at the odds of APOL1 in these 8,300 West Africans, that G2, again, was associated with this higher risk of FSGS. So what are the mechanisms of APOL1? So I won't go through the mechanisms. I'll just touch on them because Dr. Pollock really is looking at that in the lab, and lots of different people are looking at that in the lab. But there are triggers, usually, as he stated, high interferon states with an increased inflammatory states. There's probably environment that is also important. So viral infections, and there have been reports of HIV and COVID associated with worse outcomes, allograft rejection. There are what people are calling accelerators, so perhaps diabetes is an accelerator, hypertension and lupus and acute kidney injury. And then mitigators, as was talked about, this APOL1 variant that was found to actually completely negate the effect of G2 risk alleles. That was recently found. So this is, again, just a little bit of sort of what some people think is the pathology, and I'm not going to go into this too much because Dr. Pollock did, but there is basically APOL1 can affect the podocytes, and this podocyte effacement and detachment can occur. You can have glomerular injury and dysfunction. You can have increased risk of nephropathy. This can be coupled with environmental stressors that include increased inflammatory states or high interferon states that can be associated with viral infections. I'm not sure about sickle cell disease, but that makes sense. And then kidney allograft rejection, which all can coalesce into a higher risk of chronic kidney disease and accelerated end-stage renal disease. And this is a paper by Hung, who looked at APOL1 as an accelerator of acute kidney injury. So in veterans where these veterans had COVID, and she was looking at whether or not acute kidney injury was increased in those who had APOL1. And what she showed is that for the low risk group, you did have this risk of increase stage 1, 2, and 3, mainly stage 1 of acute kidney injury and COVID. But for those who had the high risk APOL1 groups, there was this increased risk of AKI. And in analyses where she looked at, she did modeling to look at what the risk was of basically the primary outcome was acute kidney injury, AKI stages, or death. What she showed is that with modeling, there was basically a 1.9-fold greater risk of acute kidney injury if you had the high-risk genotypes. There was a 2-fold greater risk of more severe AKI, and then there was a 2.15 greater risk of death in those who had the high-risk genotypes. She did this in those who had a eGFR that was greater than 60 mils and basically found the same thing. So here's another question for you. A 55-year-old African-American male with hypertension, proteinuria, 2.5 grams per day, and CKD stage 3 is found to have 2 APOL1 risk alleles. Which intervention is most likely to delay the progression of his kidney disease? And okay, so everyone done? Almost. So it looks like the answer didn't come up, but what the answer is ACEs and ARBs. And the reason it's ACEs and ARBs is that some of the newer agents, which I'll talk about, haven't actually been proven in clinical trials to be relevant yet, but they're in clinical trials. So what about ACEs and ARBs? So there are these mouse models that Dr. Pollack described that have shown that lisinopril decreases proteinuria and glomerular sclerosis while dapagliflozin did not. So there's a little bit of mouse model evidence. But the ASK study looked at ACE inhibitors and ARBs and whether or not they decreased the primary outcome. And what they found in that study when they did a secondary analysis that in 40% of those who had the high-risk variants, G1 and G2, there was actually no progression to end-stage renal disease. So there is a thought that decreasing blood pressure in these high-risk variants, individuals with high-risk variants actually can improve their progression. I could not find any data on SGLT2 inhibitors. So I think that that's an area that we need to really look at, and I'm sure that there might be papers out there that are coming out. The same is for GLP-1 inhibitors, no data. And then we know that there are some new therapies, these small molecule inhibitors that we'll discuss. I also looked and found whether there was one other study in the ASKS group where they looked at whether or not high blood pressure control actually decreased or increased survival. And what they showed is that the APOL1 group who had the high-risk variants actually had a lower risk of death compared to those who had regular blood pressure control. So the APOL1 high-risk variants had a 42% lower risk of death compared with strict blood pressure control compared to the control group, but there was no difference in cardiovascular disease outcomes. So this is one of the more recent studies inaxaplin for proteinuria kidney disease in persons with 2 APOL1 variants. So this study is one that only had, I think, initially enrolled 16 people, but then did studies on 13, and using this small molecule that basically inserted into what is thought to be the APOL1 channel actually improved disease. And what this study showed in these 13 individuals is that once they got this medication, it actually decreased proteinuria significantly, except in one individual. And you can see in the slide there that there was a significant decrease in the proteinuria. And when they looked at the mean percentage change in proteinuria or albuminuria, there was a significant decrease in albuminuria of like 47%. So this is the first time that there's been a drug that has actually shown this much of an improvement in kidney disease in this group. So I have been involved with this group, which is the KDIGO Controversies Conference, which was held in Accra, Ghana last year, where APOL1 kidney disease was discussed, looking at controversies. And so there will be more information coming from this group. But what they decided that I can share is that APOL1 kidney disease is what the group decided that maybe this whole spectrum of kidney diseases should be named for people who have APOL1, G1, and G2 variants. There are a lot of other APOL1 inhibitors in clinical development. So some are antisense oligonucleotide inhibitors that can be given by subcutaneous injection. That study has been completed. There's one that's enrolling. There is the APOL1 inhibitors, and those have gone through phase 1 and phase 2A trials, and there's a phase 2 and 3 that are now enrolling. And then there's JAK-STAT inhibitors that are also in a trial and enrolling in studies. Conclusions are that African American and Black individuals have a greater risk of kidney disease that starts early and progresses faster than other races and ethnicities, at least in the U.S. Those with African ancestry are at greatest risk for APOL1 high-risk genotypes. We know that the high-risk genotypes are associated with increased risk of albuminuria, CKD, end-stage renal disease, and rapid kidney function decline in African American individuals. And we also know from this new data from the H3 Africa study that there's a greater prevalence of high-risk APOL1 genotypes in Africa, especially in the sub-Saharan Africa, sort of West Africa region, that varies by ethnicity and is definitely associated with CKD and FSGS. That was biopsy proven. And then ACEs and ARBs are beneficial, probably slowing the progression of CKDs who have high-risk APOL1 genotypes, and they improve mortality. And we know that there are small molecule proteins that are currently being tested in clinical trials. And early data suggests that these are going to have a significant benefit in treating APOL1 kidney disease. So I wanted to just show my funding and say thank you and open it up for questions.
Very wonderful lecture. Actually, I have one question. Yeah,
yes,
sorry.
please.
Yeah, so just I'm wondering, like these APOL1 patients, we do not have any specific therapy. So is there anything like we can start early an ARB and SGLT2 together to give the more benefit to the patient than the normal patient with CKD or just a proteinuria? What is your thought about it?
Well, I think that ACEs and ARBs are probably what we could start with, whether or not adding a SGLT2 inhibitor or GLP-1 inhibitor. I think there are data out there that it improves, congestive heart failure and other diseases that are not associated with APOL1, and probably is a good thing to start, but we don't really have the evidence to back it up.
Okay. So it's similar to like normal individual we have to start. Okay. Thank you.
Thank you.
Hi, I'm Maggie Westemeyer. I'm a genetic counselor at Natera, and I just wanted to make a comment. These talks were really great and informative today, and thank you. I did see in one of the questions that folks wondered how to access genetic counseling services if your center does not have a genetics clinic or genetic counselors available to you. And I just wanted to mention that if you go to the National Society of Genetic Counselors website, which is nsgc.org, there is a find a counselor option. You can even search by specialty. Some genetic counselors may, work in nephrology clinics even and have that specialty. There are telehealth options available these days. And I'd also encourage you to speak with the laboratory who you work with, because your laboratory may also offer genetic counseling services. So thank you.
Thank you. Thank you for that.
Also, one note too, there is a handout associated with this lecture. And in that handout, I do have resources for providing counseling yourself. So like if you're interested in doing informed consent yourself, there's access to things linked out for resources, including our NSGC Find a Genetic Counselor. So that's all hyperlinked in that file. I think you should be able to access it.
And I'll open up for questions for all the panelists. Please come to the mic if you can.
Hello, everyone. Thank you for the presentation. My question is the APOL1 inhibitor that you just spoke about. So won't it affect the action of APOL1, like how it will cause side effects from completely like not forming the APOL1?
I'm going to turn that over to you.
Yeah, I'll take that. So I think obviously it's an important question with any kind of therapy, any kind of inhibitor of anything. But I think what makes this kind of an attractive candidate for an inhibitor is the fact that we know that most mammals, most animals don't even have an APOL1. So if an inhibitor is really specific in its action, we would think side effects would probably be pretty minimal, unless the exception might be unless you're in Africa and exposed to Trypanosoma brucei. But I think there's a few companies have inhibitors in trial. I think the more specific it is, the less likely it'll have side effects. But I think it's unlikely there'll be side effects due to its actual intended action.
Thank you for that question. Other questions? Well, we really, one more question.
Hi, thank you so much for the great presentation. I just had a question about the current trials being done with the small molecule inhibitors. Are they planning to have the majority of these patients on RAS blockade already, or not?
That is a good question. Martin, do you know?
I think, I'm not familiar with the precise details of all these trials. I know that at least for some of these trials, patients are continued on standard therapy, which almost always includes, ACE or ARB therapy.
All right. Well, thank you for staying over. We really appreciate it. And please fill out your evaluations. Thanks, everyone. And have a wonderful NKF session.